Hello everyone, welcome to combinatorial synthesis of azo dyes. Today we're going to be focusing on part A of the experiment because we will be synthesizing compound C3. And so here we see the TAs are preparing to weigh out 0.4914 grams of amino benzene sulfonic acid, and this is the para derivative. And so you can see here in the top right hand corner I have actually given you a screenshot of what the final mass was. So this will be something that comes in handy when it's time to determine your percent yield and theoretical yield. Now we can see the TAs are transferring their aniline or aminobenzene sulfonic acid derivative into a test tube, and now they're weighing out the sodium carbonate, and you can see here in the top right hand corner I've given you the final mass of 0.1314 grams. All right, and so they're transferring both the aminobenzene sulfonic acid and the sodium carbonate into the same test tube. And you can see they're also measuring out 5 milliliters of DI water, which they're going to add to that test tube. Right, so here's our solution of aminobenzene sulfonic acid, sodium carbonate, and water. They're adding that to a water bath that has already been on the hot plate warming. Now we can see that the TAs are going to weigh out sodium nitrite. And that final mass they got for it was 0.2016 grams. All right, and also we're seeing that they're measuring out 0.5 milliliters of water, which they're going to use to dissolve their previously weighed out sodium nitrite. Now they're adding the two solutions together, the aminobenzene sulfonic acid and sodium carbonate mixture that was warmed, and now also the smaller volume of the 0.2 grams of sodium nitrate and water. All right, now we can see that they're grabbing the concentrated hydrochloric acid, so they can get 0.53 milliliters of that and then here they're weighing out 3 grams of ice. So they're going to mix the hydrochloric acid and the ice together in another test tube, as we see right here. And so that ice water hydrochloric acid solution is then going to be mixed in with the amino benzene derivative and the mixture of the amino benzene derivative and sodium carbonate solution, and they're adding it dropwise, right? So that yellow solution, that's your amino benzene sulfonic acid, sodium carbonate, and sodium nitrite all mixed together, right? And so they're adding it dropwise to the hydrochloric acid, and they are going to now put that entire mixture together in the test tube in an ice bath. And while they're letting that cool to see precipitates, they're weighing out an appropriate mass to give them 2.6 millimolar of salicylic acid. So this is going to be on you to determine what that mass should be. All right, so they're adding that mass of salicylic acid to an Erlenmeyer flask. And now they're adding, we didn't see them measured out, but they have 2 milliliters of 2.5 molar sodium hydroxide. So they're going to dissolve the salicylic acid in the sodium hydroxide solution. All right, shaking it in order to dissolve it all. And next we're going to see them also put that in an ice bath. So there is your diazonium salt. All right, you could see a nice precipitate in there. All right, and so there is the salicylic acid and sodium hydroxide in the Erlenmeyer. It won't fit in that beaker, so they're going to get a separate beaker for an ice bath. All right, so both are in an ice bath now. All right, and they've already mixed the two together. They've already started mixing together, but they are mixing them together while stirring occasionally with a glass stir rod. And so you can see it went from kind of a yellow orangish color to now it's more of a reddish orange color. So we've already see a reaction taking place. And so what you see in this long pasture pipette, they're adding the salicylic acid to the diazonium salt solution. And they're also keeping it chilled while they do the addition, add the two solutions together. Right, and so this entire process of doing the slow addition and shaking took about 10 minutes. And so now this azo compound mixture, or this mixture to give you the azo compound, is now going to be heated on a hot plate until all of this precipitate that formed is dissolved. So while they're waiting on that to happen, they're weighing out sodium chloride. They're going to get 1 gram of sodium chloride. You can see they're already at about 0.98 grams. All right, so stopping there about 1.02. Now, all of the azo compound dissolved, so do pay attention. There has been a drastic color change. Now they add the sodium chloride to the dissolved azo compound solution, and they are putting it back on the hot plate to help it dissolve further. All right, and now at this point it is heated long enough that they're going to let it cool to room temperature, so they're just taking it off the hot plate to cool. And then next we're going to see the cooled solution. Right now they're putting it in an ice bath. All right, and once it has been in the ice bath long enough, they are waiting for that. While they were waiting for that to happen, they weighed out a watch glass and filter paper, and that picture we just saw in the top right hand corner of about 60 grams, that was the total mass of the watch glass and the filter paper. So they put the filter paper in the Buchner funnel, added their azo compound mixture so that they can filter off the solid and collect it. All right, rinsing out their reaction container so they can get everything out of it, and then they're going to wash the solid that's in the filter paper with a saturated sodium chloride solution. We're about to see them grab it. All right here, that is a saturated sodium chloride solution, and they're going to add about 2 milliliters of that to wash the solid crystals. Right, and then from there they left it on the Buchner funnel with the vacuum pump running to help dry it off. And so once it was fully dry, we now see them weighing out approximately 50 milligrams of the azo dye. All right, so not approximately, exactly 0.05 grams is 50 milligrams. Now we also see them measuring out in two 10 milliliter graduated cylinders 20 milliliters of water. So they're going to dissolve that azo compound in the water. All right, there's the water, adding the 50 milligrams of azo compound, our C3. All right, they're gonna shake it up to try and help dissolve it, but then they're also going to put it on the hot plate, heat it up a little bit to increase the solubility. Alright, so it's been heated to where it's all dissolved, so we saw a clear, translucent solution. Now they're adding cloth fabric. They're going to use the azo compound with the cloth fabric. They're going to stain that fabric, so it's going to go from looking all white to now having certain sections of it that are going to be stained distinctive colors. Right, and so they actually heat this up, keep it in there long enough, and actually get the solution to boiling so that they can adequately stain the fabric. There it is boiling. Alright, so now they're taking the cloth out after it cooled down to room temperature, and so you can see certain sections of it have been stained appropriately. Some are yellow, some are goldenrod. I mean, I dare say one of those colors might actually be orange. Alright, and so they're rinsing it off with water, and then what would normally happen in the lab is you would take the cloth that you stained with your specific azo compound and compare it to everyone else in the lab. And so here we have comparing the C3 to C1, A3, C2, B2, B1, B4. Alright, so you have all of those options in order to compare. And so you can kind of tell how most of the same segments were stained. There's slight differences between each compound in terms of what they stained. So this is an observation that you want to put in your lab report.